another way is of classifying sensory receptors by location or by type, a type of sensory stimulus that they're stimulated by. And we're going to be looking at different types of receptors shortly, but right now we're looking at the microscopic structure of the sensory neurons. And there are three types of sensory receptors. There's free nerve endings, which are bare dendrites of sensory neurons. And these would be nociception, which is pain reception, thermoreceptors, which detect heat and cold, tickle sensations, itching, and some touch receptors that we'll talk about later. And then there's the encapsulated nerve endings. These are where the dendrite is, and I forgot to look this up about the connective tissue capsule, but about whether it helps with the functioning, but it certainly does protect the dendrites. And this encapsulated nerve ending is a structure that encloses the dendrites, instead of bare dendrites. And this would be, oftentimes the name is a corpuscle, meaning the corpuscle being that capsule. So when you see corpuscle in this sense, it means a capsule around the dendrite. The term corpuscle has various meanings though. So, if you talk about corpuscles and other systems, other organ systems, it may have a slightly different meaning or a very different meaning. But Pacinian corpuscles, which are deeper pressure receptors in your skin, and Meissner's corpuscles, which are more superficial touch receptors that also detect some pressure as well, are encapsulated. And then you have a category of sensory receptors that the receptor is not in the dendrite itself, it's in a separate cell. The receptor is a separate receptor cell, and this receptor cell, when it's stimulated, it will in turn stimulate the dendrites of the sensory neuron by releasing neurotransmitters. So these are cells that produce neurotransmitters in response to stimulation, and that in turn stimulates the sensory neuron. And we'll see these in various places, like inside on the taste buds, for example, photoreceptors in the retina, and so forth. But there is and we get into those more when we get to special senses, but in the general somatic senses, it's the Merkel cells in the epidermis of the skin. And so, again, and I put a box around the dendrites up there so that you can see, I don't think my little pointer works, but in the box there, these two cells here are sensory neurons. Most sensory neurons are unipolar. Some sensory neurons in the special senses are bipolar. But either way, the dendrites are either where the sensory receptors are, or they are associated with receptor cells. But that's where sensation is, being picked up. That's where stimulation is happening in the dendrites. Once the dendrites are stimulated, they generate depolarizing graded potentials that in turn trigger action potentials in the axon, and the axon sends the signals, the action potentials, down to the synaptic end bulbs. But the actual stimulation is happening there at the dendrites of these sensory neurons. So this again is a picture just to reiterate, and we're going to be seeing these in the skin in a minute, but at the bottom is a free nerve ending. So this would be the first time in that list. This is the bare dendrites that are in the skin, sensing things like temperature, itching, by tickling, and pain. Nociceptors. In this example, this is a nociceptor. It's a pain receptor. The encapsulated ones are here and here. This is a Meissner's corpuscle of touch. That is, there's the dendrite. The little squiggle is the dendrite, and then it's encapsulated in a capsule. And here's that deep pressure Pacinian corpuscle here, and again, the dendrite is enclosed in a capsule. Once these are stimulated, they in turn generate, they generate depolarizing graded potentials, and then that can generate action potentials down the axon. The third category of separate receptor cells can be seen in the upper, more superficial part of the skin between the dermis and epidermis, You have Merkel cells. They live in the stratum basale layer of the epidermis, and when you lightly touch your skin and stimulate them, they release neurotransmitters that stimulate the dendrites, and that then can generate action potentials down the axon. So I've got a couple pictures, I think, of some of these cells we're going to come back to. So here it's showing you free nerve endings. Some examples of free nerve endings, the hair root, and again, these detect pain and itching and tickling and things like that. This hair root plexus, it's wrapped around the hair follicle. It's detecting touch in the sense of moving your hair. When your hair is moved, you're going to sense that touching. And Meissner's corpuscle, that's a touch receptor. This is the dermis, by the way. This pink layer is the dermis. The upper layer here is the epidermis. Later I'll show you a picture of the skin, and we can see all these structures in that integumentary system picture, but I don't have that up yet. So the epidermis, the outer layer, this is the dermis, and then the subcutaneous layer is where the fat is. And in the dermis, you have what are called dermal papillae, which are these little hills of connective tissue that bulge up into the epidermis, and that's where you'd find the corpuscles of touch, the Meissner's corpuscles, so that you're detecting light touch. Pacinian corpuscles are deeper in the skin, way down, sometimes penetrating into the subcutaneous layer. They're detecting deeper pressure sensations. Both of these also detect vibration. They're also vibration receptors. One detects more slow vibration, the other more fast vibration. There's also a Ruffini corpuscle. These detect primarily stretching of the skin and things like that. So they're more like stretch receptors, but they are also encapsulated. The nerve endings, the dendrites are encapsulated there as well. So that's another encapsulated one. And then we'll talk about the tactile disc. That is, I'm going to show you a picture of this. This is the one associated with the Merkel cells. So the sensory neurons dendrites are essentially bare dendrites in the dermal papillae, just under the epidermis. So let's look at that picture a little bit more detail. This is a picture from the integumentary system. This is looking at the epidermis and the different layers of the epidermis, and here's the dermis in pink. So this is the epidermis, which is epithelial tissue, and then the dermis, which is connective tissue. And in the bottom layer of the epidermis, so the top layer is up there, the bottom layer of the epidermis is the stratum basale layer, which is the basal layer of the epidermis. And that's where you find those Merkel cells, those cells that I was just talking about, those receptor cells. They actually live in the stratum basale layer. When they are stimulated by light touch, just barely touching your skin stimulates them, they release neurotransmitters that then in turn stimulate the sensory neuron. And that end of the neuron where the dendrite synapses with the Merkel cell, that's called a Merkel disc or tactile disc. Tactile means touch. So the Merkel disc is what gets stimulated, and that's just part of the dendrite of the sensory neuron there. So here's a more up-close picture. Again, notice that the Merkel disc and the sensory neuron are in the dermis. The Merkel cell, the receptor cell itself, is in the epidermis. So this cell is the Merkel cell. When it's stimulated by touch, it releases neurotransmitters, and this is a synapse right here between the Merkel cell and that tactile disc, or Merkel disc is another name for it, which looks kind of like a kind of flattened region of the dendrite where it forms that synapse. So this is just part of the dendrite of the sensory neuron. So it's got receptors for the neurotransmitter that's released here, and when this cell's stimulated, it releases those neurotransmitters, which in turn stimulate the tactile disc to generate depolarizing graded potentials, which then in turn will stimulate action potentials in the axon of the sensory neuron, and that would be down, further down here somewhere. Any questions about that? So that's a good example of a receptor cell. So we're going to be looking at the different neurons and how they generate action potentials. This one right here corresponds, if I will go, to this picture right here, the bottom picture. So they're showing you a taste receptor in your taste bud, but it could just as well be a Merkel cell. Okay, so this is the Merkel cell. If we're talking about a Merkel cell, and then this is, did I skip something? Yes, I did. Okay, that's the Merkel cell, which would correspond to this cell, and then the tactile disc is this. It's part of the dendrite. Okay, so so that would be the bottom picture. So here they're showing you free nerve endings, which would be corresponding. That's the top picture is showing a free nerve ending sensory receptor, which would be this category right here. The second picture that I'm going to show you is the encapsulated one, and then the third is the receptor cell. So this corresponds to the three types of sensory neurons. Notice that they're all, in the example here, they're all unipolar because most sensory neurons are unipolar, with the cell body off to the side, attached by a little connection to the axon. So you don't have the dendrite associated with the cell body, okay? So all of this is the axon, all that myelinated portion. Here's where the synaptic end bulbs are, right here. And this is where this neuron would synapse with a next neuron in the line, okay? Now in here it's talking about first order sensory neurons. We'll worry about that term a little bit later. But here you have the free nerve ending type of, like a touch receptor, or not a touch so much as a pain receptor or temperature receptor. So here it's showing you that this is a thermoreceptor, a cold receptor being stimulated by a cold stimulus, okay? And then here you have a Pacinian corpuscle stimulated by pressure. The dendrite is in the capsule, so that's where stimulation is happening. And then again, the receptor cell, like a Merkel cell, being stimulated by touch, and in turn releasing neurotransmitters that bind to the dendrite and generate depolarizing graded potentials. And when you get stimulation to threshold, if you depolarize the membrane to threshold, then you will generate action potentials down the neuron, to the central nervous system. So notice that the arrow is pointing in this direction. That's the direction the nerve impulses are traveling. The synaptic end bulb, these neurons are going to synapse with interneurons that then integrate that information and process it, and they then send the information to the appropriate place in the brain for perception or for processing that information. Now notice here that they're showing you what's called a generator potential. In other words, they're showing you a depolarizing graded potential in the dendrites, or a depolarizing graded potential in the receptor cell. That's what they're showing you there. We're going to talk about that in a moment. And then in the axon, you have nerve impulses, which are action potentials. These are the waves that are, you see, with action potential. So action potentials will be found in the axon. Depolarizing graded potentials are found in the receptor cells and dendrites. And it's those depolarizing graded potentials, if they reach threshold, they will then trigger action potentials down the axon. Yeah?
The Merkel cells have different graded potentials,
You mean different from the dendrites?
like different molecules.
That's a good question. I'm assuming, I have to look that up. I'm not sure if their membrane potential is any different. I'm assuming it's a similar membrane potential. In other words, the resting membrane potential is, I'm assuming -70, and then, is that right, -70, and then the threshold will be -55. But it may not be. Some other cells in the body, and muscle cells, have a different muscle cells have a -90 millivolt resting membrane potential. So it could be different, but I'm assuming because it is a sensory cell, you know, it's probably a similar, membrane potential. But I'd have to look that up, and I've never seen it mentioned one way or the other, so I'm assuming it's probably the same. So, let's see here. So you can see that they have in the little blue circles here, generator potential, receptor potential. That's what we're gonna look at next. These are the depolarizing graded potentials that are generated when a receptor is stimulated, okay? Now, so there's two kinds of graded potentials. They're both depolarizing, not hyperpolarizing, because these are stimulatory, excitatory sorts of graded potentials. So they're gonna be the wave that goes up, you know, toward threshold. So you have generator potentials and receptor potentials. If these depolarizing graded potentials reach threshold, then you'll get an action potential that'll be propagated down the axon of the sensory neuron. So let's start with, and I just put in red box, the generator potential. Generator potentials are generated in the dendrites, either in the free nerve ending type dendrites or the encapsulated ones. Either one is going to generate a generator potential. And it's just called that, and I'll show you why, but it's called that because it directly generates an action potential if it reaches threshold. The receptor potential, you get that in response to stimulating a receptor cell. So it's a potential that develops in the receptor cell, so it's called a receptor potential. And both of these, like I said, are depolarizing graded potentials. If you look at the wave, you can see it goes up. It's a depolarizing graded potential. In other words, you're starting at -70, and then when you stimulate the dendrite or the sensory cell, you open up ion channels that bring in sodium or calcium ions, and that raises the potential up to -60 or some number like that. If you get it up to -55, then that reaches threshold and you can generate an action potential at that point. So generator potentials, again, are in the free nerve endings and encapsulated nerve endings of sensory neurons. When you reach threshold, then it'll directly trigger an action potential. And that's why they're called generator potentials, because they directly generate action potentials if they reach threshold. And so that's what you have in that picture up here. Just in the dendrites, not in the axon, but the dendrites. So you get these depolarizing graded potentials called generator potentials here and here when they're stimulated by the appropriate stimulus. It's got to be, the correct stimulus. So pressure, in this case, would stimulate these generator potentials in the encapsulated nerve ending, whereas cold would stimulate those free nerve endings to generate these potentials if it were a cold receptor. So this is a cold receptor here, a pressure receptor. It has to be a specific stimulus to get those types of graded potentials. A receptor potential is produced when the receptor cell is stimulated. This triggers the release of neurotransmitter by the receptor cell. The neurotransmitter then binds to its receptors on the postsynaptic membrane, which would be the dendrite of the sensory neuron. And if you get enough inflow of sodium or calcium ions through the receptor, through that gated channel, then you'll get excitatory postsynaptic potentials in the dendrite of the sensory neuron. So let's look at that, because that's a little bit more complicated. So you stimulate like a taste receptor with a sugar molecule, or you stimulate your Merkel cells with touch. And in the case of Merkel cells with touch, you're going to open up mechanically gated channels. You're going to let in, like sodium ions or some kind of positive ion. That's going to depolarize this membrane of this cell, and that depolarization triggers the release of neurotransmitters. So here's a bunch of synaptic vesicles like you would find in a synaptic end bulb. So these cells are just, they're little cells. They don't have axons and dendrites, but they have the ability to release neurotransmitter. So they release the neurotransmitter, and that neurotransmitter travels across the synapse, binds to the receptors for that neurotransmitter. That neurotransmitter opens up, that receptor opens up, it's a ligand-gated channel, so it opens up, inflows sodium ions, and that's going to depolarize the dendrite. What they don't show you here, which they should really, is you get another depolarization, but this depolarization we call an excitatory postsynaptic potential, or EPSP. It's just, it's like any other postsynaptic potential. When you open up the receptor for the neurotransmitter, when that gate opens and sodium flows in, that's going to depolarize the membrane, and if you depolarize it to threshold, then you will generate action potentials in the axon all the way down to the synaptic end bulbs, and then these synaptic end bulbs are going to release neurotransmitter in response to that action potential. So, in all of these cases, when that action potential arrives, that stimulates the release of neurotransmitter, which in turn stimulates an interneuron in the central nervous system, and and then that sensory information gets propagated to the appropriate place in the brain. This picture is showing you, here's an action potential which happens in the axon, in the trigger zone of the axon, but to get this in the axon, you have to generate a strong enough depolarization, a strong enough generator or receptor potential to get to threshold. So in the case of in 1, 2, and 3, those represent either receptor potentials or generator potentials. And so if you have a weak stimulation, let's say of a free nerve ending or something like that, you're going to open up a few ion channels, sodium flows in, you get a little bit of depolarization, but it's sub-threshold. And number 2, it's a little bit stronger stimulation, but it's still sub-threshold. But when you hit, I guess it's 3 or 4, I'm not sure, 4 is threshold, but anyway, when you reach that third line here, that is a threshold stimulus, and that's enough stimulation to generate an action potential in the axon. So you have to either summate, or summate by weaker stimulation, or having more and more stimulation, but weak stimulation, but more and more of it, summating to threshold, or you can have a very strong stimulation that gets you to threshold. Either way, to get to threshold, to generate an action potential, you have to hit threshold, which is about negative 55 millivolts. Keep in mind that when you look at receptor potentials, there's a receptor potential, that's what stimulates release of neurotransmitter. The neurotransmitter then generates EPSP. So in the case of receptor potentials, you could also look at this as EPSPs in the dendrite of that sensory neuron. In other words, the neurotransmitter released by the receptor cell opens up, you open up gates, inflows sodium, and if you get enough sodium flowing in and you reach threshold, you'll generate an action potential in the sensory neuron axon. So this is, again, just going with this. Oops, let's see here. So, sub-threshold 1 and 2, I guess, would be similar to this kind of stimulation where you just get a little blip, but you haven't hit threshold yet. Whereas number 3, I'm not sure what corresponds with what, but anyway, I'm assuming 3, you've hit threshold, 3 or 4, you've hit threshold, that would be analogous to this one. Threshold stimulus just enough to get you to threshold to generate an action potential. Negative 55 millivolts is threshold generally. Suprathreshold, if you're stimulating the neuron a lot, if you're stimulating that sensory receptor a lot, whether it's generator potential where you're getting more depolarizing graded potentials, higher depolarizing graded potentials, or if you're stimulating the receptor cell and it's releasing a lot of neurotransmitter, which is opening up a lot of gates and allowing a lot of sodium in. If it's suprathreshold, remember that action potentials, you don't get bigger ones, you just get more of them. And so the more stimulation there is, if it's above threshold, you'll generate not just one action potential, but lots of them flowing down the axon. And your brain, when it receives this information from these sensory neurons, it interprets, just a few action potentials as being a weak stimulus, and it interprets lots of action potentials as a stronger stimulus, and it integrates all that and gives you that sensation that's appropriate to that stimulation. So when you're just barely touching your skin, you can tell you're just barely touching the skin versus when you're really putting pressure on the skin. It's different kinds of stimulation. Your brain's interpreting that based on how much information is coming in by way of those sensory neurons. And that's what, we talked about before. You can put a gram of weight on your skin and stimulate those pressure receptors in your skin. 1 gram is just barely hitting threshold. Just once in a while, you might summate it to threshold and get an occasional blip, which is an action potential. Those little vertical lines are action potentials that are being measured. And so, you can see that there are periods of time where there's not enough stimulation, where there's no action potentials, but, occasionally that 1 gram might stimulate it enough to get it to threshold, so you get occasional blips. And your brain is going to interpret that as barely any pressure at all. It may not even really, you may not even be consciously aware of that sensation. It's so weak. But then, the more weight you add to that skin, and the more weight you put on those pressure receptors, the more gates are going to open, allowing sodium to flow in, the more depolarization, more, in the case of Pacinian corpuscles, we're talking about generator potentials. So the more generator potentials are generated, and the stronger the generator potentials, the more action potentials you're going to get. And so your brain is going to interpret all this data basically as very strong stimulation versus weak, and the various levels of stimulation. So your brain takes that information and interprets that so that you perceive that sensation. Any questions about that? Now in here somewhere, I thought I had a thing about mechanically gated channels, so maybe that's later. I thought I had it in here though. Okay, maybe I lost that picture, but anyway, no, maybe it's later. I'm thinking in the later picture, but I thought I had it here. So anyway, let's move on to adaptation. Adaptation refers to the perception of a stimulus. You might notice that sometimes a stimulus, or the feeling that you get from a stimulus, gradually fades away. That, when you first put on your clothes, you notice the clothes on your skin, but after a while you don't notice it anymore. Or, if you smell like somebody around you smokes cigarettes, and you can smell the cigarettes on their clothes, but they can't smell the cigarettes. You're being freshly stimulated with that smell, and you notice the smell, but they have been around that smell so much they've adapted to it. And smell is one that you strongly adapt to very quickly. Like when you're working in a restaurant, you may not notice the smell of the food in the restaurant, but people who come into the restaurant notice it. So you become desensitized. So really adaptation refers to becoming desensitized over time when you have constant, unchanging stimulation. Now if the stimulation changes, if it becomes stronger, then you'll notice it again. So, your clothes lightly touching your skin, you may not notice after a while, but if you put on another layer of clothing and add more clothing, or if you move around and move your clothes on your skin, then you notice that feeling again. So you become, desensitized for a while until that stimulus changes. So it's the perception of stimulus gradually fading or disappearing under constant, unchanged stimulation. If the stimulation changes though, then you'll notice that stimulus again. So what happens is during adaptation, as neurons, some neurons do adapt very quickly, some don't, but those that adapt quickly, you see a decrease in the amplitude in receptor and generator potentials, which results in a decrease in the number of action potentials that are being generated and propagated. So as a result, there's a decrease in sensitivity. Yes?
Okay, so when people become overstimulated or and
does that mean there's like a problem with this, or is it due to something else?
What do you mean overstimulated? You mean people who are easily overstimulated in terms of behavioral?
No, it's like they
things?
don't, we were learning about it in nursing. It's like,
Sensory?
sensory overload.
Oh, like with autism?
Yes. Like that.
Sensory overload. Well, I don't know, I'm not sure, I don't know the context of it, so I'm not sure, but, would it be, like, in what context are you talking sensory? You mean just by normal stimulation, they get overstimulated, or by too much stimulation coming in?
Well, it was like sensory overload was like when people couldn't shut out certain
Oh.
stimuli, and like the light was overstimulating them, and like the
Yeah,
sounds.
so some
and-
people have trouble, their brain has trouble kind of filtering all the stimulation coming in. Most people can kind of filter out a lot of stuff and focus. You can focus on what's at hand. Well, there are some people that have, because just the way their brain works, they may be more easily distracted by stimulation and not able to filter out. That's kind of the brain perception part of it, is filtering out certain stimuli that aren't, you can kind of say, oh, that's not important right now. They have a harder time of doing that. But exactly why that is, I couldn't tell you. I'm not a psychology major, but yes.
So like, if your skin was raw and you touched it with the same amount of pressure you touched a different part of your skin, and you would like feel the rawness more, is it because the nerves are exposed?
Well, pain reception that's one of the things that doesn't adapt well. So it turns out there's actually slow adaptation and fast adaptation. And that's what we're going to talk about next. But pain is a slow to adapt one, whereas touch is more of a fast adaptation, or certain types of touch. I don't know if I answered your question, and I have to look into it more, but I don't know if I answered your question or not. But
A for effort.
this is really more at the sensory neuron level, not so much the brain level, but it probably does play a role in what your brain can deal with, because maybe part of it is due to a lack of ability to adapt. Maybe their neurons don't adapt as well, and if that's the case, then you're getting more constant stimulation that other people wouldn't get. Does that make sense? Like touch reception and things like that oftentimes are fast adapting, and if your neurons don't adapt, then you're going to feel that longer, and it's going to be more distracting for longer periods of time, if that makes any sense. So it could be at the sensory neuron level, but I don't know for sure. Yes.
One question.
Yeah.
Okay, so does different reactions to touch and feeling happen because of different types of neurons? Like where your fingertips are more sensitive than, say, like your arm?
Right, we're going to talk about that. That has to do with receptor fields. And so you have more, like your fingertips and other places have more sensitivity because there's more neurons there. And other places are more spread out. And so you have more sensitivity in some places than others. And that goes for other things like cold and, heat reception and stuff like that. So that's a good question. We're going to get to that, I think, today. But anyway, we'll talk about adaptation first, and then I think that's coming up pretty soon. So as I said, there's two kinds of adaptation. There's slow adaptation, and there's rapidly adapting receptors. So slowly adapting receptors, what are called tonic receptors, respond at a constant rate to a stimulus. They adapt very slowly. In other words, they take a while to generate fewer action potentials. And sometimes they don't adapt at all. You have some of these, what they call slowly adapting receptors. They really ought to be called not adapting, because many of these slowly adapting receptors, or tonic receptors, have such low adaptation that they don't, in some cases, adapt at all. And this would include pain. As I said, pain, oftentimes you don't have adaptation to pain. If you've got like raw nerve endings, or that are from an abrasion, or any other pain source, sore muscles and things like that, it just keeps on hurting, which is why, pain can be so draining on a person, because if you're in constant pain and it doesn't adapt, it can, it affects your whole life. But other things can also be slow adapting. Body position or proprioception is slow adapting. and so is chemical composition of the blood, which is a good thing because you don't want to adapt to high CO2 levels. You don't want if you have high CO2 levels, you don't want your neurons being less stimulated by that. You want them to be like right on top of it, and since chemical composition is constantly changing and fluctuating anyway, there wouldn't be much time for adaptation. But still, you don't want sensory neurons that are detecting CO2 levels and pH of your blood and things like that. You don't want them adapting to that. You want them to be constantly monitoring and sending the information to the brain as that stimulation comes in. And so there's a picture here that shows slow adapting versus fast adapting. Here they apply the stimulus in this example, stimulating the neuron, stimulating the production of either receptor or generator potentials, which in turn stimulate action potentials. And you can see those action potentials just keep getting generated pretty much at the same rate. If you let it go a while longer, maybe you would start seeing the action potential spread out, but it might take a while. It's going to be slow adapting. In this case, it really hasn't adapted at all. And some of those slow adapting fibers or receptors, and then we're talking about the receptors because that's where the stimulation is happening. Slow adapting receptors sometimes never adapt. Like I said, with pain, you often don't have any adaptation at all. The rapidly adapting receptors, which are also called phasic receptors, they respond initially with strong, stimulation. With any stimulation, they respond at first with generating lots of action potentials, but then those over time, they become less sensitive and they generate fewer depolarizing graded potentials, which results in fewer action potentials. And that's really what adaptation is certain neurons adapt to constant stimulation, others really don't adapt at all, or very little. So slow adapting, they don't adapt very well. The rapidly adapting adapt quickly. And a lot of touch receptors, pressure receptors, and smell receptors are that type. Also temperature receptors are rapidly adapting. For example, when you first get into a swimming pool and it feels really cold at first, or in the ocean or wherever you are, you're swimming, and at first it's cold, and then after a while it feels like you're in, room temperature water, or body temperature water. After a while, you don't notice it being cold anymore. It's not that the water temperature changed, it's still cold, it's that your cold receptors have adapted. And so instead of being, stimulated initially, they're stimulated, and let's say it's, cold reception. You're in the swimming pool at first, you're generating lots of generator potentials, which generate a lot of action potentials. But then over time, those receptors become less sensitive and generate fewer generator potentials, which generates fewer action potentials. And so your brain at first interprets it as really cold, but now it starts to slow down, those action potentials slow down, and after a while you don't even notice the cold anymore. It doesn't feel cold anymore. And so that would be an example. Or smell reception. At first, you smell the food being cooked, you smell it strongly, then after a while it becomes less strong and after a while you don't really notice the smell at all. So smell reception is another rapidly adapting type of or fast adapting receptor. Any questions about that? So even though they call those top ones slow adapting or tonic receptors, they are sometimes not adapting at all. It depends on the type of receptor though. Some of them adapt, but they adapt slowly. Okay, so any questions about that before we move on? Now we're going to look at somatic receptors, which are sensory receptors in the skin. In other words, cutaneous receptors, muscles, tendons, and joints. And as we were talking about in terms of sensitivity, there is an uneven distribution of these receptors. Some places in your body have a high concentration of certain types of sensory receptors. Other places have fewer receptors. So places where you have more receptors, it makes sense you're going to be more sensitive. So like tactile or touch, you're very sensitive in your fingers and in your face, but you're not so sensitive in your arms and legs. But cold reception, you might notice when cold water hits your back, you can really feel it. But then other places like your arms, it's not as cold feeling on your arms as it is your back. So some places have more of certain types of receptors and fewer in other places. And so it means you're going to be less sensitive in some places, more sensitive in other places. There are 4 what we call modalities of somatic sensation. In other words, 4 categories of sensation. Modality just means the type of stimulus. So 4 categories of somatic sensation. Tactile, which means basically touch, but it also includes pressure, vibration, itching, and tickling. Thermal or temperature, meaning hot and cold. Pain, and proprioception. Those are all what we consider to be somatic sensations. So first of all, the tactile sensations, there's lots of them. We're going to look at the different examples: touch, pressure, vibration, itching, and tickling. And we're going to look at these different types. There's Meissner's corpuscles, as I mentioned, which are touch receptors. And they also respond not only to touch, but pressure and slow vibration. Hair root plexuses, which are receptors surrounding the hair follicle and detect movement of hair. The Merkel discs, which are those cells in the dermis that are stimulated by Merkel cells. Ruffini corpuscles, which are in the dermis and detect stretching of the skin, but also can be found at ligaments and tendons and detect stretching of ligaments and tendons. Pacinian corpuscles are pressure and vibration receptors. They're found in the skin and subcutaneous layer, but they're also found in some internal organs. We're focusing on the skin, but some of these receptors, like Pacinian and Ruffini corpuscles, are also found internally as well, inside the body. And then there's, of course, the free nerve endings that detect, in terms of tactile sensations, they detect itching and tickling. They do detect pain, but that's a separate category. So we're just looking at the itching and tickling part. Okay, so this picture shows you the different types in the skin. I'm going to show you an overview of the skin and kind of point those pictures out as well there. But the top portion is, find something to point with. This is the epidermis. Remember that stratified squamous epithelium, and that's the surface of your skin. The deeper dermis, which is connective tissue, very vascular and full of blood, full of sensory receptors, and then the subcutaneous layer, where the fatty layer is between your skin and your muscles. And so you can see the different types. We're going to come back to all these different types, but what's nice about this picture is it describes each type as well as points them out. So we're going to kind of go through each type one at a time. And there's another picture that just shows the type name without all the information. So either picture, it could be useful for studying. If you're trying to memorize them just by name, you can use that picture. If you're trying to learn the parts or what they do, you can use that picture. So, first of all, in the tactile reception, and this is the picture I was wanting to see earlier, in tactile reception, in the sensory receptors that detect touch. So this would be, the Merkel cells would have these kind of mechanically gated channels. The touch receptors, like Meissner's corpuscles. The pressure receptors, like Pacinian corpuscles. The receptors that detect vibration, which are Pacinian and Meissner's corpuscles. The stretch receptors. All of those receptors that are stimulated by some kind of what we call mechanical stimulus, which is touch, pressure, vibration, stretching, things like that. Those are all mechanical types of stimulus, as opposed to chemical stimulus or some other type of stimulus. In the receptor itself, so either in the dendrites, if it's a free nerve ending or encapsulated nerve ending, or in the receptor cell, if it's that type, like the Merkel cell, they have these mechanically gated channels in their plasma membrane. It's closed when the gate is closed. These are protein gates, and the gate is closed when there's no stimulation. But when it is stimulated by, either the touch, pressure, stretching, vibration, that kind of thing, it's going to open up, and in flows sodium ions. So usually sodium, it could be, I suppose, calcium ions, but it's a positive ion flowing in. That's going to depolarize the membrane, it's gonna generate either a receptor potential in a receptor cell, if it's a Merkel cell, or a generator potential in the dendrite of the sensory neuron, you know, that's where the sensory receptors are. So this is what does the reception right here. It's the stimulation of these mechanically gated channels. Now, not all receptors work this way, as we'll see when we get into some of the other types of receptors, particularly in the special sense organs. There's other ways that these receptor potentials or generator potentials happen. But in most tactile sensation, with the exception of itching is a different thing. But in terms of vibration, tickling, and touch and pressure, and I think I said vibration and all that, those kinds of mechanical types of activities open up the mechanically gated channel in the sensory receptor and inflows sodium ions, depolarizing it and generating either a generator potential or a receptor potential, which in turn can generate an action potential on the axon. So that's how those kinds of receptors typically work. All right, so let's look at the different kinds. Meissner's corpuscles are also called corpuscles of touch. They respond to light touch. They also respond to slow vibration, like if you put your hand on a vibrating car, you know, with the motor running, you know, that's slow vibration. And they also respond to pressure, to light pressure. So just barely, you know, touching the skin, that's light pressure. So touch and pressure are almost the same thing, really, in that sense. But they have a connective tissue capsule, it's kind of egg-shaped connective tissue capsule around the dendrites, where the sensory receptors are. They are rapidly adapting. So these generate nerve impulses at the onset of touch, but after a while, you know, like if you were to, you know, touch your hand and like do this over and over, you're just stimulating them. But if I put my finger on my hand and left it there, after a while I wouldn't notice my finger was resting there. I wouldn't even notice it, wouldn't feel it. I would become less sensitive. So it's fast adapting. That's why when you're wearing your clothes, usually unless the clothes are heavy or rough or itchy or something else, you don't really notice your clothes because they're just, you know, they're stimulating those Meissner's corpuscles, which then rapidly adapt to that sensation. But when you move, then you re-stimulate those receptors or stimulate other receptors in your skin, and then you notice it. So when you're moving, you notice your clothes moving on your body, but when you're sitting still, after a while, you don't really notice it. And just sit there and think about it. What do you feel in terms of your clothes? Some of the places you may feel if there's pressure and it's pushing on your pressure receptors, but other places where it's looser, you may not even notice the clothing on your skin. Now you find these in the dermal papillae of hairless skin. I'm not sure if I'll show you a picture of the skin picture. I should have put it in earlier. But here's where the Meissner's corpuscles are. This is the dermal papillae. These are those little bulges of connective tissue into the epidermis region. That's a Meissner's corpuscle. The dendrite is inside there. And then the axon would be here. And that axon would then enter a nerve and, you know, carry information to your spinal cord or to your brain. If it's in your face, it would go through like your facial nerve to your, or other cranial nerves to your brain. But that sensory neuron, that axon is here. So the dendrite is here, the axon is here. And some of the places where you find, and by the way, it's found in the dermal papillae of hairless skin. So I forgot to mention that. Hairless skin, in other words, skin like on your lips, fingertips, hands, you know, the palms of your hands and your fingertips, the soles of your feet and toes, eyelids, tip of the tongue, lips, nipples, clitoris, and the tip of the penis. Those are places that are very sensitive, don't have any hair, they're going to have Meissner's corpuscles. So Meissner's corpuscles are found more in those kinds of locations. And again, that's there. So it's way up close to the epidermis, and so it's going to be very light touch. It's going to stimulate those things. Another one, the hair root plexus, is the next one we're gonna look at. And the line doesn't quite hit the neuron, I just realized, but this thing that's wrapped around the hair follicle, remember hair follicles surround the root of the hair, that's where hair grows, is the hair follicle. And it's surrounded by these free nerve endings. Basically, hair root plexuses are nerve networks or dendrites that are wrapped around the hair follicle, and what they sense is hair movement. So when the hair is blown by the wind, or if, like say, an insect is walking on your skin and tickling those hairs, you would feel that. That's one of the benefits of having hair on your body, is that you can feel if an insect, like a tick or something, is walking on you, so you can knock it off. Without hair, a little insect is so light, it may not even stimulate the Meissner's, well, not the Meissner's corpuscles, but the other touch receptors in your skin may not get stimulated. These are rapidly adapting, so after a while your hair's blowing around, but after a while you don't really notice it anymore. So it's, you know, you become desensitized fairly rapidly in these types of receptors. Any questions about that before we move on? Okay, so Merkel cells. Merkel cells are actually, like I said, we talked about these, so let's go back to the pictures and then we'll come back to it. This picture I should have put in earlier, but I was doing this really quick this morning. This is that picture from the integumentary chapter: epidermis, dermis, subcutaneous layer, and I'm just going to quickly point out all the neurons that you can see, sensory neurons you can see. So Meissner's corpuscle would be here. That's a Meissner's corpuscle. That's that one we just talked about, touch receptor. Free nerve endings that detect itching and heat and cold, that's a free nerve ending. These are also found in the dermal papillae of the dermis, in these little bulging areas. And the term dermal papilla, here's if you saw them without the epidermis, they just look like these little bulges like that. So free nerve endings are found there. Pacinian corpuscles deeper down. And what I want to point out is the tactile disc. So if you take this and magnify it, that's what you're getting here, is a magnified image of the epidermis. And again, the stratum basale is where you find the Merkel cells, which, when stimulated, release neurotransmitter stimulating the Merkel disc of the sensory neuron, in other words, the dendrite. And there's that picture again, we just talked about it, so I'm going to move back to this. Anyway, Merkel discs, which are the sensory neuron part of the receptor, or these are, the Merkel cells are actually the receptor cells, but these are the cells that receive that information from the Merkel cells. The Merkel disc, or tactile disc, is in the dermal papillae. The Merkel cells are in the stratum basale layer of the epidermis, so right there at the junction between epidermis and dermis. These are also found in abundance in your fingertips, hands, lips, and genitals. And again, these are places where you're the most sensitive. These are slowly adapting. They're not fast. So some touch receptors are slow adapting. These are slow adapting. So these you would stimulate, and they would continue to be stimulated fairly consistently. It would take them a while to become desensitized. So if you're feeling, that your clothing on your body, putting pressure, touching your skin, what you're probably being stimulated by over, if it's been a while and you're still feeling that sensation, it's probably those Merkel cells and Merkel discs that are being stimulated. Any questions about that? Now, when you look at the picture that's in your book of this, this is what it really, the Merkel disc looks like. What they show here is, there's that, and they're also called tactile discs. The tactile discs are at the very tips here. They almost look like synaptic end bulbs in that picture, but they're not. They're the tactile discs. What you cannot see are the little bitty Merkel cells in the epidermis, but there are, associated with each disc, there's going to be a Merkel cell that's going to be stimulating that tactile disc, and then, in turn, generating EPSPs here that generate action potentials in the axon. And then there's another just a picture without the description. Any questions about any of these so far? Okay. Ruffini corpuscles, that's what this one is right here. They're kind of in the middle of the dermis. They are also encapsulated. They are also found in tendons and ligaments, and we're going to get into that a little bit later. But these are slow adapting. So they are stimulated by stretching, and they take a while to adapt to that stretching. They are, so not only stretching of the skin, but stretching of tendons and ligaments during movement. But in terms of tactile sensation, we're just talking about the skin here. So they're in your hands and in your soles of your feet. When you're stretching your hands or you're walking and you're stretching the soles of your feet, they are being stimulated. And this helps you monitor how your skin is being stretched, whether it's being overly stretched by movement, that kind of thing. So they help you kind of just monitor movement of the body by monitoring not only your tendons and ligaments stretching, but even your skin and how it's stretching. But particularly, you've got them throughout your body, but they're especially abundant in the hands and soles of the feet. Okay. So there it is, Ruffini corpuscle. It also says it senses crude touch, but I think it's most important in the stretching of skin. Okay, so Pacinian corpuscles, they're the ones that are way deep down. They're very large, deep inside the skin. Looks like an onion, layers of onion around the dendrite. Though they are connective tissue capsules there. So these guys are involved in sensing deeper pressure. So if you put more pressure on your skin, you're going to stimulate them. They also are sensitive to fast vibration. So if something's vibrating really fast, they're going to get stimulated deep down in the skin. Maybe because slower vibration doesn't really get down into the skin like fast vibration does. They are fast adapting, so after a while they become desensitized with the same stimulation. They are found in the dermis, deeper in the dermis than some of these other receptors. They're also found in the subcutaneous layer, and they are found in deeper structures within the body. They're found around joints, tendons, and muscles. They're found in the periosteum of bones, which is the outer membrane around bones. So if there's pressure put on bones, they're going to be stimulated. They're found in other places as well, mammary glands, external genitals, and some internal organs. For instance, the pancreas, of all things, has these large Pacinian corpuscles in it. So if there's any kind of internal pressure being put, like say you were being crushed by something, something was putting a lot of pressure on your body, they would be stimulated. So it helps kind of monitor pressure internally as well as at the surface of the body. So there's an example there. Free nerve endings, those again are the ones that sense itching and tickling. They're important in other sensations as well, but itching and tickling in terms of tactile sensation. And that's what they're showing here. Again, free nerve endings are going to be in the upper portion of the dermis, like in those dermal papillae, not deep down in the skin. Now, those are all tactile receptors. Any questions about any of the tactile receptors? Okay, so we're going to look next at the free nerve endings, which detect pain, heat, cold, as well as itching and tickling, which itching and tickling are actually, tactile. But anyway, so let's look at the free nerve endings involved with thermal sensations. I should take out itching and tickling, you just want to remember as part of tactile sensation. But anyway, thermal sensations. These are free nerve endings in the skin that respond to either heat or cold. They are fast adapting. Like I said, if you get into a cold bath or shower or swimming pool, you adapt fairly quickly. At first it's painful and cold, and then after a while you go, oh, it's fine now. But then you get out of the water for a while, and for a few minutes, and you get back in the water, and it's just as cold as it was before, because now you're re-stimulating those neurons, re-stimulating those receptors, and regenerating waves of action potentials. But the longer you stay in the water, the more you adjust to it. Cold receptors are activated between 10 and 40 degrees C, or 50 to 105 degrees Fahrenheit. They're at 105 sounds high, but it's not really in terms of temperature reception in the skin. There's more cold receptors than there are warm receptors, which is probably why when you get in cold water it's so shocking, because you have more of those cold receptors. Warm receptors are activated. By activated, that's when they are stimulated at 32 to 48 degrees, or 90 to 118 degrees Fahrenheit. So you can see there's some overlap in temperature, but they're going to be activated, and they're going to create send signals to your brain. Your brain's going to put that information together, and if there's more cold stimulation, then you're going to interpret it as colder temperature. If there's more information coming from warm receptors, your brain's going to detect that you're warmer. So it's the different degrees of heat and cold, or it's your brain putting it all together and coming up with that. Yeah.
It's kind of a curious question. Since like body temperature doesn't normally go over 100 degrees, how is a cold receptor activated at 105?
That's a good question. I don't know why it's activated at that temperature.
It just seems like, how would you know, if it's not...
I know. That's a good question, and because 37 degrees Celsius is body temperature.
Yeah.
So it may be, though, when it says activated, and I have to look into this because I'm curious about that too.
You'd think anything above body temperature would just feel warm.
I know. I think what it is, though, is the number of action potentials that are generated vary with temperature, and when your brain receives that information, it's like data processing. It's interpreting the number of action potentials coming from cold receptors and heat receptors and putting it together as a sensation of cold, as a perception of, if it's more coming from warm, then you're going to have that sensation that you're warmer. More coming from the cold, you're going to have that sensation of cold. So I think it's a combination of the two. In your brain, it's perceived as a certain temperature. Does that make any sense?
I guess
So
like they're
a
both
lot of-
firing off, but just you don't feel it as much as you do the warm ones.
Right. The warm's probably generating more action potentials at that higher temperature versus cold generating fewer. Is what I'm guessing, but I'd have to look into that and make sure I'm telling you right. Now, if the temperature's too hot or too cold, it can be perceived as pain, because then you're starting to stimulate pain receptors. So when you burn your hand, or your hand gets really cold, ice, from ice, that is actually stimulation of pain receptors as well as the cold receptors. Yeah.
What happens when you go numb?
When what?
You go numb?
Go numb from cold?
Yeah.
I think that has to do, well, that's a good question. I think it has to do with blood supply to the area, and that affects the nerves, the neurons functioning. So, it depends. I mean, if it's cold from numbing cold, what happens is your hands, when you're that cold, your blood vessels constrict and send blood to your core to keep it warmer. But that process also makes your skin number, loses sensation, because I think you have a lack of blood flow. So those neurons are kind of shutting down their activity because of a lack of blood supply. But I'd have to make sure that I'm not telling you wrong, but I'm pretty sure that's what's happening, is that lack of blood supply affects the neuron function. And it's, as long as it doesn't happen too long, it doesn't affect the neuron function. It could, though, frostbite and that kind of thing could affect it, definitely affect it, killing tissue and all that. So, but yeah, that would be that case of numbness. Now, if it's numbness from another reason, it could be nerve damage or spinal cord damage or something like that. But in the case of heat or cold temperatures, it's just, I think, a lack of blood supply. Same thing like when your arm goes to sleep. You sleep with your arm like that, and then you wake up, your arm, because it's been in a funny position, it's numb, and you've lost all sensation, you've lost blood flow to that limb, and those neurons are not firing like they should be until they get a blood supply back. Okay, let me see what else. I was going to say too, you do have different amounts of these receptors in different parts of the body, so some places are more cold sensitive or heat sensitive. But if you want to test how fast adapting these fibers are, you can do a test like take cold water with ice in it, put your hand in there, and just see, you know, time how long it takes for your hand to adjust that temperature. And same thing, you can do your hand in hot water and see how long it takes for it to adapt to that hot water and where it starts to just feel like normal temperature. Because they will adapt fairly. You don't want to burn yourself or freeze yourself, but you can, you know, test yourself with mildly warm and mildly cold water and see how, just with your hand, and see how quickly it adapts. Okay, pain. Any questions about thermal reception before we go on? Okay, so you have pain reception. This is also a free nerve ending. As I mentioned, they're called nociceptors, and they, you know, warn you about tissue damage. It's, you know, an adaptation to prevent you from further damaging a structure, or, you know, it allows you to know you've injured a structure so you can treat the structure and keep from re-injuring it. Free nerve endings are pretty much everywhere in the body, including internal organs, with the exception of the brain, which is why they can do brain surgery without knocking you out, without general anesthesia. And in fact, they prefer not to give you general anesthesia, because general anesthesia can have side effects. Plus, when you're doing brain surgery, you want to make sure the person is still functioning, still able to, you know, still alive, still, you know, able to respond to instructions. You want to make sure you're not doing any brain damage. You know, so they might have you blink your eyes or do something to make sure everything's working properly. So they can monitor your brain function while they're doing brain surgery. This is also how they've been able to map out the functions of the brain by stimulating different parts of the brain during brain surgery. They've been able to find out a lot about brain function by doing that. So fortunately, we don't have brain or pain receptors in the brain, so that can happen. Lots of things can activate nociceptors, as we were talking about, intense heat or cold. If it's too hot or too cold, it can stimulate pain receptors. Mechanical things would be things like abrasion and puncture wounds. That's mechanical stimulation. And then chemical stimulation would be things like if you poured acid on your hand, or a strong base on your hand, and it would burn your skin. That would be chemical stimulation. So certain chemicals are, you know, can activate nociceptors and create pain. Also, when you have like inflammation or damaged tissue from an injury, that injured tissue releases chemicals such as prostaglandins. Prostaglandins are also able to stimulate nociceptors and create pain. So some pain comes from stimulation by chemicals released in your own body. For example, another place you can see prostaglandin secretion is in the uterus during a woman's menstrual period. Many of you women know that, you know, at the beginning of your period you have pain from having your period. That pain is due to prostaglandin production, which is kind of high at the beginning of your period. That stimulates pain receptors and stimulates, you know, it stimulates some smooth muscle contraction in the uterus. It also stimulates pain, which is why women have pain during their period, but also pain, you know, menstrual pain, but also pain during having a baby. Those prostaglandins stimulate nociceptors, and that creates pain during labor contractions. Nociceptors exhibit very little and sometimes no adaptation. In other words, the pain just keeps on hurting and doesn't seem to get worse or get better. Sometimes it decreases, but it usually decreases as either the structure heals, or, you know, if a scab forms over the free nerve endings, or something, you know, happens to block some of that stimulation. But oftentimes the pain can go on and on and on, requiring pain medication. Okay, so there's two types of pain. There's fast and slow pain. Look at the time, make sure I have time for this. Fast pain happens in your skin. It happens like in response to a puncture wound, like if you get a needle prick or a cactus prick, or you burn your finger, or, you know, it's usually some kind of, you know, injury to the skin kind of thing. And it creates sharp, prickling pain. This type of pain is where a certain type of nociceptor is stimulated that it has myelinated axons, and those myelinated axons transmit that information very quickly to the brain, so it is interpreted as very sharp and very localized pain. You know, everybody's experienced fast pain. It's called fast pain because it is sent to the brain very quickly, so you feel it very quickly. When you prick your finger, it happens fast, you feel it fast, because the impulses are traveling down myelinated axons to the brain. Not felt in deeper tissues of the body. So if you get a puncture wound into the deeper tissues, you're going to stimulate a different kind of pain, which is slow pain. Slow pain is felt a little bit after the stimulation because it travels through unmyelinated fibers. So the nociceptors that are stimulated have unmyelinated fibers. This slows down impulse transmission, and so the brain receives this information a little more slowly. So, you know, you stub your toe, for example, and initially it doesn't hurt, then all of a sudden it hurts, you know, and so, or you have some kind of other type of slow pain. It's usually like muscle pain, you know, throbbing pain, you stub your toe, or you do something like that. It's oftentimes chronic, and it gets worse sometimes over time. So as your brain starts receiving those signals, the pain perception gets worse. So initially it may not hurt too much, but then over time it hurts more and more, like when you stub your toe. At first it may not hurt so much, and then it gets worse and worse. That's slow pain. And slow pain occurs not only at the skin surface, but deeper tissues in the body. And that's the kind of pain you have that's internal. Internal pain is slow pain. Wait a second, very quickly. We're going to talk about next time. We're going to begin with referred pain. That's where we'll start. So study up to this point in your notes. That's what the side clicker will be on. We'll finish this topic on